So last week Monday, I'm sorry, which last week? Monday. We finished the conversation with a discussion of lymphatic system. We figured that the main components are the lymph nodes, that filter. You have assemblages of the lymph nodes like in the groin, in the cervical area, an axillary area, appendages, assemblages that can be palpated fairly easily. They're close to the skin. And a lot of lymphatic vessels, from smallest capillaries to collecting vessels to trunks and ducts. And the point is that we made is that they are very much like veins. Thin-walled, they have valves, they have a lot of anastomosis, and as we, basically just as veins, one of the main mechanisms for pumping lymph are respiratory and muscle pumps. Same deal. Valves prevent the backflow of lymph. And since a lot of lymphatic vessels run along the major arteries, like thoracic duct runs along the aorta, the pulsatile pulsations of those large arteries help to propel the lymph. Does that make sense? Now, collecting vessels, there are many. The parts that I can ask you on the anatomy exam, which is not going to be on Monday, remember, not going to be on Monday, we're going to merge it with respiratory, are some of the trunks and ducts. Okay? So you've got paired lumbar trunks, you've got unpaired intestinal, you have paired jugular trunks, you have subclavian trunks, also paired, and bronchomediastinal trunks. By location, you can fairly easily figure out which one's which. Basically, the jugular trunk runs along the jugular vein. Subclavian trunk runs along the subclavian vein. Bronchomediastinal trunk runs along the bronchomediastinal vein. Okay? The only difference is intestinal, which is kind of sort of runs along the rectal artery, rectal vein, and you can see that lumbar trunks, sorry, they run more along the iliac veins or iliac arteries. Does that make sense? But that's anatomy-wise. I can ask you the trunks. Okay. Now the ducts. So I have thoracic duct, which collects the lymph from everything except the right arm and the right side of the head and the thorax. So this duct collects the lymph from lower limbs, from the abdomen, from the left arm, left thorax, left side of the head. Does that make sense? That makes sense? And there's a right lymphatic duct, which is fairly short. It's right here. Right lymphatic duct collects lymph from the right side of the head and right arm, right side of the thorax. That makes sense. Now that lymph ends up in the blood and it enters the venous circulation at the junction between the jugular and subclavian veins on each side. So right lymphatic duct, it collects the lymph and delivers it to the junction between the right jugular and right subclavian. Thoracic duct delivers the lymph into the junction between the left subclavian and left jugular. Does that make sense? Anatomically, it's it. Good? That's all we need to know about now. Now let's talk a little bit about lymph, lymphatic vessels, and structures of the lymph nodes. First, we already figured how lymph is moving. Okay. Basically, if you think about lymphatic system, you can imagine it as sort of a railroad network where vessels are railroads and individual stations are represented by the lymph nodes or spleen in some cases. So lymph moves from one station to another, one station to another. And cells of the lymphatic system are present mostly in the lymph nodes. Tissue in the lymph nodes is loose reticular connective. Okay, so the cells that produce reticular fibers, you can see them here, reticular cells. These reticular cells produce fibers that form some kind of a mesh. Inside of this mesh, you're going to find cells of the lymphatic system, immune cells, such as lymphocytes or macrophages. Does it make sense that I really, you need to know what cells are in the lymphatic system? So we've got lymphocytes and macrophages that are clinging to the reticular fibers produced by the reticular cells. Does it make sense? Obviously, this mesh of reticular fibers works as a filter. Does that make sense? And we'll figure out how lymph flows through the lymph nodes and through the spleen. There are two types of lymphoid tissue: diffuse lymphoid tissue and follicular lymphoid tissue. Diffuse lymphoid tissue is the structures of lymphatic tissue, such as reticular fibers, or reticular cells, or lymphocytes, or macrophages, distributed sort of evenly across the tissue. They do not form any specific package. Does it make sense? So cells that are lymphatic cells that are interspersed around other lymphatic cells. You can find it in, let's say, lamina propria diffusely around the gastrointestinal tract. Okay? That makes sense. So if you would take the intestines from an average person, and you would make a section of the tissue, and you would make specifically a section of lamina propria, a very thin slice, and stick it under the microscope, you're going to be able to see lymphocytes there. They do not form any specific structure. They are just in that connective tissue, areolar connective tissue, which is lamina propria. Alternative version of lymphatic tissue is the follicular lymphatic tissue. Forms follicles, associations, structured assemblages of the lymphatic cells. And it can be found in the lymph nodes, Beer's patches, tonsils. That's basically it. Okay? That makes sense. So we're going to talk about the lymph node as the main organ of the lymphatic tissue. When lymph flows from lymphatic capillaries to the collecting vessels, to the trunks, and eventually to the ducts, it encounters the bean-shaped lymph node. If you're curious about the size of a typical lymph node, it's about the size of a pea. It's not really big. And if you would look at the shape, there's a very clear zone here known as hilum. Hilum is the place where so-called efferent lymphatic vessels drain the lymph from the lymph node. Afferent lymphatic vessels, shown here. This picture you can see 5 afferent lymphatic vessels. They deliver lymph to the lymph node. Now you may remember terms afferent and efferent from nervous system. In peripheral nervous system, afferent fibers are sensory that go away from the central nervous system, and efferent fibers are motor that go towards. Okay, so same analogy. Efferent vessels go to the lymph node, afferent vessels leave the lymph node. Am I clear here? Is there any confusion? I hope not. Now I want you to tell me the numbers that you see on this picture. How many efferent vessels are there? Efferent, oh, efferent too, sorry, yes. And afferent. 5. So there's an obvious mismatch. You have 5 pipes that bring the lymph to the lymph node. You have 2 pipes that drain the lymph away from it. What does that mean from the standpoint of lymph in the lymph node? Is it going to be like cleared rapidly? It's like a pool, right? If you have a drain open in the pool, but you have a hose that pours water in there, and if you have like 5 hoses and only 1 drain, there's a good chance that the pool will overflow. Well, the lymph node is not overflowing, but that sort of a mismatch that allows lymph to stay inside the lymph node for an extended period of time. Does that make sense to you, this particular statement about the hydrodynamics? Okay, so here's the deal. Lymph node contains a lot of immune cells, from macrophages to T cells to B cells. In order to recognize the potential pathogen and mount the response to that pathogen, the exposure takes time. When lymph is somewhat stagnant in the lymph node, you have this time. Are we clear? We're good. Okay. So there are specific zones in the lymph node. The outer cortex and the inner medulla. They're not clearly anatomically separated. So outer cortex, which may say something like here, okay, contains so-called germinal centers with B cells. This is where B cells undergo clonal selection and then clonal expansion. And when you have a lot of plasma cells formed in the lymph nodes, they spill over into the lymph and eventually end up in the blood. Does that make sense? That's where they produce antibodies. Okay? In the deep cortex, closer to the medulla, you're going to find T cells. And in the medulla, right here, you're going to find T cells, B cells, and plasma cells. And this makes a lot of sense from anatomy and physiology standpoint. Because cells, as they mature, and you have to understand, when we say, hear me out, when we say B cells become plasma cells, yeah, right, there are like 6 steps, which I totally do not remember. Like, I'm not a hardcore immunologist. But there are like several steps which B cells go through. And as they go through the steps, they move closer and closer from cortex to the medulla, closer and closer to the hilum. Because that's where they're going to go. They're going to enter the efferent vessels, so lymph can bring them into the circulation. Does that make sense? Okay? So in medulla you can find T cells, B cells, and plasma cells. In the medullary sinuses, the tiny lymphatic capillaries that run through the lymph node, you're going to find macrophages. Okay, but I do not focus that much on location of macrophages. What I want you to understand is that there is a fairly clear separation of B cells being mostly in cortex, T cells being mostly in the medulla. Was that clear? Yeah, kind of boring part structurally. Each lymph node is encapsulated into the layer of connective tissue called capsule, and extensions of capsule called trabeculae, they partition the lymph node. So far? Yes?
I just wonder for diagnostic testing, do they ever aspirate lymph?
I am not sure about lymph aspiration because it's easier to draw blood, but for diagnostic purposes, you can do a biopsy of a lymph node. And even in case of infectious diseases, and we'll talk about it, it makes sense to biopsy an enlarged lymph node to see if there is any pathogen in there. But I don't know about lymph aspiration. Really. Blood is just easier because it's going to be there anyway. Now, spleen. The largest organ of the lymphatic system. Let me see. Here, sort of, it's a fragment of it. Okay, easy. Here, spleen, left hypochondriac region. Okay. The main function, it does immune responses. It does immune surveillance. It stores a lot of white blood cells, T cells predominantly. In case of infection, those T cells get released. A lot of memory T cells are stored in the spleen. This is actually one of the very elegant, although labor-intensive methods to determine if anyone has ever had T cell response to an infection. What you technically can do, you can take a person's splenic biopsy, grind it up, isolate T cells from the spleen. It's doable. It's a technical issue, absolutely technical issue. And then you can take those T cells and expose them to the pathogen. Ideally, they should be mixed with dendritic cells, so there's presentation. Does that make sense? And then T cells, if they are exposed to the pathogen that they saw before, they just start to grow. In humans, it's hard because it's hard to convince the patient to give a spleen biopsy for this experiment. There are less traumatic methods, but in mice, in order to see if mice produce T cells to a particular pathogen, that's the common way. Of course, mice do not get any chance for biopsy. You just sacrifice the mouse and take the entire spleen. It destroys old cells of the blood. Okay. Since iron is precious, spleen also stores iron. This partially explains the color of the spleen. It's also highly vascularized organ. Okay, a lot of blood vessels. And another interesting feature about spleen, which I actually kind of, in some experiments, I saw it myself. There's a lot of cell division going on all the time. Does that make sense? So if you take a sample of a muscle and you isolate RNA from it, you're going to get, let's say, 50 nanograms of RNA for 1 gram of tissue. You take spleen, you can get like 800 nanograms of RNA. A lot of cell division, a lot of gene expression. It is very, very active tissue. Okay? Basically, you can think of spleen as a really, really big lymph node. Enormous lymph node. It has capsule, it has trabeculae. It has 2 types of tissue, though. The red and the white pulp. Red pulp mostly stores red blood cells and releases them in case they're needed. White pulp, obviously, stores mostly white blood cells and releases them when they're needed. Does that make sense? And since there's a lot of exchange between the blood and the splenic tissue, it contains a lot of sinusoid capillaries. So old, worn out red blood cells can get into the splenic tissue, and new red blood cells or lymphocytes can enter the circulation. Sinusoids are really permeable. Does that make sense? So here on this image you can see the artery and the vein, and then those are flat, permeable sinusoids. Well, on this I think you can see the red and white pulp. Okay, is it clear? Now a few words about lymph nodes and spleen and diagnostic importance and clinical relevance. Often when you get infection, like a respiratory infection, you have enlarged lymph nodes. It's a local response to the infection, say in the throat. Local response involves proliferation of B and T cells. Lymph nodes becomes bigger. Does that make sense? If you have, and generally this condition can and its inflammation can often manifest as inflammation, enlargement, swelling of the lymph nodes. It can be an inflammation of the lymph nodes. It can resolve after the infection is gone, or if you have some tricky bug, it can stay in the lymph nodes for a really long time. Like adenoviruses, sure thing. In this case, people sometimes develop a condition that is called lymphadenitis, chronically inflamed and painful lymph nodes. Now, sometimes the infection can spread from the local lymph node along the lymphatic vessels. So in the patient with this condition, you can see red streaks under the skin, inflamed lymphatic vessels. It's very common in subcutaneous infections with microbes like Staphylococcus aureus or Streptococcus pyogenes. This condition is called lymphangitis. Lymphatic angitis, inflammation of vessels. Angio refers to vessels. Does that make sense? Another interesting feature, now not about lymph nodes, but about spleen. Ever heard the word splenomegaly? What is that? Enlargement of the spleen. It is often associated with various types of chronic infections. Since I claimed it on multiple occasions, I'm a microbiology guy. I love various types of microbes. Condition known as malaria causes very prominent splenomegaly. One of the diagnostic features, because Plasmodium falciparum, the pathological agent, it infects red blood cells, and they become essentially defective, and spleen tries to get rid of them and becomes super active and stores a lot of iron, and all the activity leads to the enlargement of the spleen during the malaria. And spleen actually is critically important. It's so interesting that just a little clinical tidbit. Have you heard about the infection called babesiosis? If you ever donated blood, you should. Babesiosis? No? It's a protozoa. It's like malaria, same type of protozoa, unicellular eukaryotic organism. But the difference with malaria is that it is found in the United States. You can get it basically anywhere in the Northeast. And it's transmitted by ticks. So people who have spleen, if they are infected with Babesia, they're going to have like fever, like bad fever. They can be treated, they're going to be fine, they're going to be healthy, everything's going to be okay. That makes sense? They will never be able to donate blood though, but they're going to be okay. People without spleen, if they're infected with Babesia, they most likely will die. Because spleen stores T cells. For some reason, it's spleen. It's T cells, that is the only way for the immune system to get rid of Babesia. And the drugs that we have, they cannot completely clear the infection. So without the help from the immune system, people are doomed. Okay? So, however, I have to highlight, spleen is not an organ that is necessary for life. Can be removed. It is often removed in people who suffer different types of blood malignancies. Lymphomas, leukemias. Think about this. If there is, especially in lymphoma, if spleen starts to produce malignant white blood cells, you want to get rid of an organ that produces malignantly, white blood cells. You don't want to keep it there. So often spleen is removed in this case. Does that make sense? Last few things to talk about in terms of the lymphatic system. Thymus, or fork-shaped gland, a little above the heart, very active during childhood. The only function of a thymus is to make sure that T cells mature properly. Okay? It shrinks as we grow older and loses its function. However, even if you will test people who are really, really old, you will still be able to see some residual activity, some residual T cell maturation in the thymus, although it's mostly gone. And by gone, I mean it physically shrinks. It becomes smaller and smaller and smaller. Now, few important things about thymus. Yeah, it does have cortex, outer cortex, it does have inner medulla. What I want to highlight first, there are no B cells in thymus ever, only T cells. That's a very distinctive feature. Second, T cells, maturing T cells, lymphocytes, they are isolated from the blood in the thymus by a so-called blood-thymus barrier. And I want to talk about it with you a little bit because it's so exciting, super exciting. So T cells in thymus, they mature. Basically, they learn to recognize between two types of cells. Which types? T cells. What they must not attack. Any immune cells. What they must not attack. Our own cells. Self, right? So in thymus, T cells learn what is the difference, basically, between self-antigens and foreign antigens. We're clear. Thymus, during this maturation, well, essentially what they learn what self-antigens are. So anything that they will be exposed when they mature in thymus will be considered as the self-antigen. Does that make sense? Do you understand? It's like when you train a dog and you tell the dog that dog learns, here's the family. And if you have a friend of a family that comes to your house very, very often, and dog will likely treat that friend of a family as the actual family. Does that make sense? Okay? So same goes for T lymphocytes. They must be completely isolated from any possible pathogens. So they do not develop tolerance to them. Guess what? In mice, for one pathogen was shown, it's so-called murine memory tumor virus. Basically, it's a virus that causes breast cancer in mice. Okay? This virus sneaks into the thymus, and it basically infects the thymus, so that T lymphocytes that mature in thymus, they recognize the antigens of this virus as pollen, which I think is marvelous, just absolutely marvelous. Think about it. Just absolutely, it's a great, great mechanism. So again, lymphocytes, when they mature, they do not come in contact with any pathogens whatsoever, and there are no B cells in thymus. Mucosa-associated lymphoid tissue. Lamina propria across the body. Most famous arrangements of mucosa-associated lymphoid tissue are in the intestinal walls. So you can see here, follicle. Lymphoid follicles contain B cells, T cells. They're not as structured as lymph nodes. They're still pretty well structured. Okay? These follicles are basically the first responders to potential pathogens that enter from the intestinal lumen. Does that make sense? Okay? And that mucosa-associated lymphoid tissue includes structures like tonsils. They will respond to pathogens coming from with the respiratory activities, with the air, or Bear's patches. Bear's patches, you can see the follicles in the Bear's patches that can be found in the lamina propria of the large intestine mostly. So this is the lumen of intestine. Those are Bear's patches. I'm going to show you Bear's patches. So you can see that Bear's patches are distributed all around the intestine. There's a lot of Bear's patches around appendix. Okay? These follicles, they just protect against pathogens. If they cross the mucosa and try to sneak through lamina propria, they will produce B cells and T cells and will build immune response before it's too late. Does that make sense? Okay. I think we're done with lymphatics. It's really, really tiny, really, really small. Any questions on lymphatics?